So analysis and mechanisms. This topic is related to all organic chemistry reactions and their mechanism. So you should be able to distinguish, like when it's an alkane, it will undergo free radical substitution reaction. If it's an alkene, then it will undergo electrophilic addition reaction. If it is halogenoalkane, it will undergo nucleophilic substitution reaction. So you should be able to identify electrophile and nucleophile and draw the correct arrows as well. So first, whenever we have an organic compound, we first try to identify certain characteristic of the organic compound. One of them is identifying its molecular mass. So how to identify a molecular mass in a mass spectrum? The stages in the mass spectrum, as we already discussed in the unit 1, that first what we do, we vaporize the sample, then we ionize the sample, then accelerate, then deflect, and then detect. So using the mass spectrum, we obtain these peaks. So the one which is the highest mass to charge ratio, that is known as the molecular ion peak. And that mass is actually a molecular mass. Like if we are getting at 58, so it means 58 is a molecular mass. But we are getting other peaks as well, like 29 and 43. And one which is the highest percentage abundance, that peak is known as the base peak. So there's a difference between the molecular ion peak and base peak. Molecular ion peak, the one which is having the highest mass to charge, whereas base peak is the one which is having the highest percentage abundance. How to identify the number of the fragments which are produced in the mass spectrometer? So different combinations you will make. So identifying the number of the fragments in mass spectrometer. So how we identify the number of the fragments in the mass spectrometer? There are different fragments can be produced. Like example, if you have a molecule such as butane, example. So butane, when I draw the structure of a butane, it means it will be CH3, then it will be CH2, then it will be CH2, and then it will be CH3. This is butane. Now, how to identify the number of the fragments? So you will break, what you will do to identify the fragments, you will break from left to right, and then you will break from right to left. So we break this molecule. So if I break, I may get CH3. That is one possibility. So where we'll get a peak? Carbon is one carbon and three hydrogen. So carbon is 12 and hydrogen is 1. So 12 plus 3. So we'll get a peak at 15. So there will be a peak at 15. Then you might have CH3 and CH2. So where we'll get a peak? 2 carbon, so 24 plus 5 hydrogen, so 24 plus 5, so we'll get a peak at 29. You might get 3 carbon, CH3, CH2, CH2. So till here it was 29, 29 plus 14, so you might get a peak at 43. And the peak, the fragments, whenever you're mentioning the fragments with their peak, you should always write the chart, because ions can be accelerated in the mass spectrometer. That's why the fragments with a charge can be detected. So then we'll get a peak at 58. So these are the peaks if we break from left to right. And if we break from right to left, so what will be the peaks? So this will be CH3 15. Then it might be CH3 CH2, so 29. It might be CH3 CH2 CH2 43, or it might be CH3 CH2 CH2 and CH3. So it will be 58. So we are getting the common peaks because the molecule is symmetrical. So the total, sometimes they ask total number of the peaks. So how many peaks you will get? You will get a peak at 15. You'll get a peak 29. You will pay 43. Common one we write always at once. So we'll get a peak at 15, 29, 43 and 58. Is it clear? The first example, then fragments, how to identify the number of the peaks in the mass spectrometer when the molecule break down into fragments. So another example, say we have aldehyde, CH3 CH2 C double bonded with O NH. So how to identify the number of the peaks? So break from left to right. So there will be a peak of CH3. It means there will be a peak at 15. Then there will be a peak at CH3 CH2. So 24 plus 5, so 29 will be there. And there will be peak of this all. Like carbon, you always break from carbon, not with other. So CH3, CH2 and C double bonded with ONH. So that is when you get the full molecule, you call that as a molecular ion peak. So till here it is 29, 29 plus 12, so it will make 41, and 41 plus 16, so it is 57, and plus 1 it is 58. So we'll get at 58. And when we break from right to left, so it will be CHO peak. So carbon is 12 plus oxygen is 16, 28 plus 1, 29 will be there. Then it will be C double bonded with ONH, and then CH2 will be there. So 29 plus 14, so we'll get at 43. And then the last one, that is again the molecular ion peak will be there. So it is CH2 and CH3 will get at 58. So what are the peaks we'll get? 15, 29, it can be for this one or the other one. So 29 is common. And then there will be 43. And at the last, it will be 51. So these are the 4 peaks in the mass spectrometer of, this is 3 carbon. So it is propanol. So in propanol, we'll get 4 peaks, 15, 29, 43 and 58. And if we have propanone, so if there is propanone, so CH3, C double bonded with O and CH3. So break from left to right and then from right to left. So when you break from left to right, left side we have CH3, then we have CH3, C double bonded with O, and then we have CH3, C double bonded with O and CH3. So we'll get peak at 15, 15 plus 12, so that is 27, and 27 plus 16, so that is 43. So we'll get 43 and this will be 58. And same way, if we break from right to left, because it's a symmetrical molecule, so it will be 15. Then CH3, C double bonded with O, that will be 43. And CH3, C double bonded with O and CH3, that will be 58. So the peaks which we are getting 15, we are getting 43, and we are getting 58. So where you can clearly see that propanol and propanol, they have the same molecular mass. This is 58, and the previous example, that was also 58. So they both have the same molecular mass. So how the propanol mass spectrum of a propanol will be different from mass spectrum of propanol? There will be four peaks: 15, 29, 43, 58, and there will be three peaks: 15, 43, and 58. And the peak which can be used to distinguish whether it is a propanol or propanol, and that is a peak at 29, which is due to either C double bonded with ONH or CH3CH2. So that's a missing peak which is not present. This peak is not present in propanol, but it is present in propanol. So sometime they give you this common question, that they give you the mass spectrum and they ask which peak can be used to distinguish between propanol and propanol. So how we can identify? We can work out the number of the fragments it break down. And as you can see in this example, this will remain same. So always the 29 is always present in propanol, but it will not be there in propanol. Is it clear, this part? Same way, sometimes you have like different types of alcohols and you have to identify the number of the peaks. So CH3, CHOH, just break from the side of the carbon, because there are many complex results can be there, but you are taking the simplest one. So if we break from left to right, so we might get a peak at CH3, we might get a peak at CH3 and CHOH. So what is attached, that will not break, only will break the bond between the carbon atom. And don't forget to put the positive charge, because why always positive? Because when we ionize the sample, we target them by a fast moving electron. So the electron knock out the other electron, so electron is being removed. That's why there should always be a positive charge on a molecule. Then we might get a peak at CH3, CHOH and CH2, or we might get a peak at CH3, CHOH, CH2, CH3. So this will be, because CH3 12 plus 3 15. What about this one? 15 is there already. 15 plus 12, that is 27. 27 plus 16, that is 43. And 43 plus 2, 45. So we'll get peak at 45. Then 45 till here, it is already 45. For 45 plus 14, so we'll get at 59. And 59 plus 15, because already till CH3 CH2 we already did, so that will make it 74. And if we break from the right hand side, from right to left, so we'll get a peak at CH3, CH3 CH2, CH3 CH2, CHOH. So already 29 plus this remaining part. So it is the same thing, CH3, because the other side CH3, so this will be 59. And then 59 plus the remaining CH3, so it will give Always don't forget to put the positive charge because this is having a mark that it shows that this is a molecular ion, a positive ion, so this will get 74. So what are the total peaks we'll get? The total we are getting 15, we are getting 29, we are getting 45, we are getting 59 and 74. So this is a secondary alcohol or butane to all. We are getting this. These are the peaks for butane to all. But what if it was butane one all? Like OH is attached with the first carbon, then what will be the difference? So OH is there, CH2, 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 and CH3. So we break from left to right, and we break from right to, so left side to right hand side, it will be OH and CH2, or it will be OH, CH2, CH2, or it will be OH, CH2, 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 or it will be OH, CH2, 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 CH3. And from right to left, so it will be CH3, or it will be CH3, CH2, or it might be CH3, CH2, CH2, or it might be CH3, CH2, 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 and then OH. So what will be the calculation? Like this is 16 plus 1, 17, and this is 14. So 17 plus 14, that will give 31. So till here 31 is there. 31 plus 14, this will be 45, and 45 plus 14, 59, and 59 plus 14, 59 plus 15, sorry, because this is CH3. So the molecular ion peak will be there 74. What about this one? This is 15 because CH3 is there, so 15. So 15 plus 14, that is 29. Then 29 plus 14, that gives 43. And then the molecule, whole molecule is there. So the peaks which we are getting, 15 is there, 15 is there, 29 is there, 43 is there, 45 is there, 59 is there, and then 74. So if you want to distinguish whether it is primary alcohol, which is example butane 1-all, or it is butane 2-all, so we can distinguish from this that 15 is there. So 15 can, we will check, like here this is having 15, the other is also having 15. So 15 cannot be used to distinguish because we want to identify which peaks can be used to distinguish whether it is butane 1-all or 2-all. Then 29 is here, 29 is there, so 29 cannot be used to distinguish. 45, 45 is there, cannot be used to distinguish. 59, 59 is there, so cannot be used to distinguish. 74, that's a molecular, and because these are isotopes, these are isomers, so they have the same molecular mass. So only 43 is there, which can be used to distinguish whether it is butane 1-all or butane 2-all. So it is important that you work out the different fragments and identify their mass, and according to their mass, you can work out the number of the fragments or the fragments which can be used to distinguish between the different types of compounds. So these are the examples like the mass spectrum of propanol and propanol. Any question or a doubt till this point? So we started this analysis and mechanism. First, we are doing the analysis part, like how we can identify the molecule, trace the molecule, and the later part we will discuss different types of mechanism, which we already did in unit 1 and 2, but that's like a revision for each mechanism. Any question or a doubt related to this session today?